be working on your exams. And in fact, in this case, I am the one solely grading the exams, having some more house issues, bottom line. So I had to babysit my home, took them all home with me, had a little party grading. It was like, I probably graded for 10 hours yesterday, and I would guess I'm only halfway there. A lot of work. So my hope is that you'll see your grades on Friday, Saturday at the latest. And then we'll go over the exam. If you want to see your exam and you can't come to office hours, next Tuesday or this Tuesday, stay after class, we'll do our regular thing. Or you can go to office hours. From now on, office hours is going to be in that 2251 room. We don't have to go to the third floor anymore. And I'll be there today, but we're not covering the exam. So don't try. Don't come trying to cover the exam. What did I want to say? I know I have one more announcement. Yeah, maybe it'll come to me. I remember, so Mirari came by, I said that right?
Yeah.
Mirari just came by and she's like, hey, is there extra credit on the final? And I said, no. And she's like, hey, what gives? And I said, well, it's going to be the easiest exam you guys have had so far. There's 40 questions. It's because it's multiple choice, you can't help but have some questions that really are just brute force memorization, which I don't love, but there's no way around it. There will be several questions on the exam that are in the spirit of what you've been doing, right? You'll just have 4 or 5 answers to choose from, and then there will be some calculations too. But I promised Mirari, and it really is true, that that it's a short exam. You're going to get 2 hours for it. It will feel short. It is open book, okay? You know, the only thing is that if any of you guys try to take advantage of the situation and do any kind of shenanigans, there are powerful tools afforded to us on Canvas. I've caught students who their wrong answers correlated. The semester didn't end well for them, right? So just don't do it. Don't take the chance. It's not worth it. It's going to be doable for you guys, I promise. All of you are ready. And by the way, the other thing I told Mirari, she said, well, wait a minute. How about all of us who have been showing up? You know, the room is now crazy empty. You know, I mean, I think this is like COVID safe at this point, maybe. But, you know, she's like, what about all of us who've been showing up? And it's like, I look across this room. I don't know the names of every single one of you, but I bet you if I went through, I'd be pretty close, right? So I know who's showing up. I know who's trying hard. And so don't worry about it. You know, when it becomes time for that extra little bit that you might have gotten for extra credit will be made up for. When I'm determining final grades and I see someone who's been showing up to lecture over and over and over consistently, that's what's going to make the difference, for sure. All right, so let's start doing some stuff. Do some biochemistry. So we're really, I believe we have 6 more lectures, and then that's it. You guys will be officially done with me forever, ever, and ever. And so let's just finish strong, right? These are really cool chapters. You know, chapters 16, 17, and 18 are, in some ways, the best, because now we're going to apply all of that big brain knowledge that all of you have to understanding these really neat biological questions, culminating in what, in my opinion, is one of the greatest discoveries in the history of science, the molecular mechanism for how ATP is generated. So I know you guys are excited. That's why you came today. I'm excited too. So we're going to start with glycolysis. And, you know, so the name of the chapter is glycolysis and gluconeogenesis. There's a good reason why they include gluconeogenesis here. So glycolysis, of course, the processing of glucose into a final product called pyruvate. And these are a series of 10 very important reactions, but they're not the end-all be-all of energy production. In fact, it really is just going to be the beginning of energy production. As we'll see, once you perform glycolysis, it's really the next steps that are going to be the more important in terms of, or the more direct, I should say, in terms of generating energy, generating ATP. But that being said, it is true that our bodies have to be able to generate glucose at times. Like when we all go to sleep at night, this organ right here loves glucose. And so our bodies all have the ability to, when glucose levels get low, our bodies will generate glucose. And so that's why they put it in this chapter, even though it is different, and we will, the most important take-home message is that gluconeogenesis isn't simply the reverse of glycolysis, and all of you should know why. If glycolysis is energetically favorable, then going the opposite direction is not favorable, and there's going to have to be the coupling of an energetically favorable process to at least some of those reactions. So it is different, but again, I think it does make sense to include a brief discussion of gluconeogenesis when covering glycolysis because, again, it's important, and a lot of the reactions are going to be the glycolysis reactions in reverse, okay? But we'll spend 95% of our time on glycolysis. And depending on if I start rambling too much, I may just ask you to cover gluconeogenesis on your own. We'll see. Okay. So, as I alluded to, what we're going to be covering in the next 6 lectures is glycolysis, which is shown here, the citric acid cycle, which is shown here, and then a process called oxidative phosphorylation. Oxidative, what does that mean? Well, we already kind of know now, we've covered that the energy to make ATP ultimately is going to come from the favorable oxidation of fuel molecules. The oxidation of sugar, for instance, releases a ton of energy, right? And that energy somehow is going to be stored, harnessed, for the generation of ATP. And that's really where we're going. And by the way, glycolysis will end up generating some ATP. And it actually generates another important molecule called NADH, which we discussed a little bit already in chapter 15. Right, and so that's going to happen. We're going to get pyruvate. Eventually that pyruvate is going to go to an organelle called mitochondria, where ultimately ATP synthesis is going to occur. That NADH is converted into acetyl-CoA, another molecule that we discussed in chapter 15. We said this is a special molecule for 2 reasons. One is that it has what we call an activated bond. It's an activated carrier. The thioester bond between coenzyme A and the acetyl group that's ultimately coming from glucose, that's a bond that can do work. Right? The hydrolysis of that thioester bond is going to end up being useful, and we're going to see why. Right? And we're going to see how enzymes are able to harness that energy at the molecular level. But then there's that acetyl group. And if you guys forget what an acetyl group is, it's a good idea to draw this. Wake up. I've had a stickier than me today. Here's an acetyl group. So does this thing have some oxidative power? I'm not sure that's the right way to put it, but a better way to put it is, can we say, does this molecule have some high energy electrons that can do work? The answer is yes, because this methyl group right here has 3 carbon-hydrogen bonds. And the oxidation of each one of those is going to release energy. And so, as we'll see in chapter 17, the processing, I should just say the oxidation of acetyl-CoA in the citric acid cycle is going to generate NADH and a close cousin of NADH called FADH2. So effectively, those molecules are just taking those high-energy electrons and storing them. And then ultimately in the mitochondria, the oxidation of NADH and FADH2 is going to generate that proton gradient that we talked about. And that was like Lake Mead, right? It's effectively a lake of protons that run through a molecular turbine that I'm going to show you in beautiful, exquisite detail, not only how that works, but some of the experiments that were used that were pivotal in determining that molecular mechanism. In fact, I had the great fortune of, when I was doing my PhD at UCLA, one of the guys who co-discovered the mechanism of ATP synthase, named Paul Boyer, was awarded the Nobel Prize. And it was a big, big deal. You know, we had a ceremony, and there were over 1,000 people in the courtyard by the building. And I remember, I'll always remember, it's like goosebump moments when he was very old at the time. I think he had to have been maybe already in his late 70s, but maybe 80s. He's an old guy. And I just remember like the look on his face, and it was pretty cool. The world, when you get to be my age, you just get more and more jaded by the world. But there's always going to be these beautiful moments, no matter how jaded you get. And so we're going to look at Paul's work, and we're going to understand how is it that this enzyme is able to drive an unfavorable reaction. How is the energy stored in that proton gradient transduced into solving this problem? You guys are going to love it. So glycolysis is a metabolic pathway. There's going to be 10 reactions that we're going to study. And so the reaction is going to be converting glucose into pyruvate, and then we're also going to generate 2 ATP. We're also going to generate 2 NADH. That means on my left side, I need to have 2 ADP plus 2 PI, 2 NAD plus. It's an anaerobic process. So that means that this pathway can run in the absence of oxygen, right? And so we're going to see in chapter 18 why we breathe. And that's a good thing, obviously. But there are times where we can do things, impressive things, in the absence of oxygen. Really good, my favorite example is when I think about rock climbing. When you're a rock climber and you're trying hard and things are getting grim, these things right here start to get super, super painful. And your fingers start opening up and you're like, oh my God, I'm about to fall 30 feet, it's terrifying. And what happens is, the fact that you can keep going under that duress is because the muscles in your shoulders and your back and your forearms that climbers are using can go anaerobic for a period of time and still generate ATP thanks to glycolysis. So even though, as we discussed, I absolutely, most of us cannot hold our breath for really more than a minute before we're in big, big trouble. So obviously, to sustain our lives, we need oxygen. But there are parts of our bodies that can go anaerobic for significant periods of time, thanks to glycolysis. Okay, so when we're well fed, meaning when we have access to a normal diet, it's fair to say that glucose is a really, really important fuel. it is true to say that under conditions where you're actively, there are people who have, there's all kinds of crazy diets out there, right? Like the ketogenic diet, where it's like all you eat is fat and protein, and by the way, those people are still going to have some glucose, because as we'll see, fat is converted to glucose, right? So no matter what they do, no matter how hard they fight, their bodies are still going to be producing this stuff to some extent. But when we consume, hey Shalji, you can come up here. It would be weird having you back there. When our bodies have access to sugar, your brain wants to use sugar, and so to me that tells me something. It tells me, I'm not so convinced that it's maybe a good idea to completely eschew sugar, and it's delicious. Right, 10 reactions in common. And this is an evolutionarily conserved pathway, by the way. You can't really find, there are organisms that are obligate, like viruses. If the biologists in the room can correct me, I don't even know if there's probably still a debate on whether people even consider a virus an organism, a living organism or not. But if you do, clearly you don't, but if you do consider it a living organism, but there are also things like parasites, and they rely on another organism. So we can all basically agree on that. If you don't have to rely on another organism, then you are performing these reactions. These reactions are as old as life. And once again, we're actually going to break this thing up into 2 stages, right? We're going to, it's a little different than our chymotrypsin's 2 stages, but we're going to break this up into stage 1 and stage 2, and what we're going to see is that in stage 1, we're actually going to invest ATP. And so immediately you should be thinking to yourselves, well, why the heck would we utilize the thing that we're trying to make? And the answer should be intuitive to you, because it's going to set up glycolysis ultimately to generate what we need to generate ATP. Not only in stage 2, where we'll make up that ATP and then generate extra ATP on top of that, but ultimately we're trying to get set up to have the big payoff when we get to oxidative phosphorylation. All right, so let's just go through now the reactions one by one. And, do you need to memorize the drawing of this thing? No. Should you look at that thing? I mean, if I look at that molecule, I would not be able to tell you definitively that that's glucose. I just know it's a sugar. Okay, that's good enough. If I give you the numbers of the carbons, that's going to end up being useful, as you'll see. And eventually, you'll see that once we deal with these open chain forms, the numbering is going to be totally intuitive. All right, so step number 1 is going to be the phosphorylation of glucose, generating glucose 6-phosphate. So that's going to require an ATP, right? So we're investing in ATP, generating glucose 6-phosphate. Now, once again, there are good reasons for putting that phosphate on there, and we're going to talk about that. I don't want to get into it just yet. Right now, I just want to go over the steps. And then this reaction is catalyzed by an enzyme called hexokinase. So the word kinase should immediately tell you this is an enzyme that catalyzes phosphoryl transfer from ATP to the substrate. The next step is going to be catalyzed by an isomerase, which is going to turn glucose 6-phosphate into fructose 6-phosphate. And so, I don't know, if I look at this thing, I go, okay, well, yeah, it does look different. Look at that. So I go from a 6-membered ring to a 5-membered ring. The phosphate hasn't changed positions. That's still on the 6th carbon. That's why we call it fructose 6-phosphate. The 6 tells me which carbon the phosphate is on. And like I said, that's going to be really helpful for us, keeping track of stuff. Now, once again, they have done an injustice for our arrows. Like that. Implying that these are not completely irreversible, of course. Once we have fructose 6-phosphate, there's going to be a really, really important reaction by an enzyme called phosphofructokinase. This is going to be another phosphorylation event, this time on the OH group on carbon 1. It's going to generate fructose 1,6-bisphosphate. And so this phosphofructokinase ends up being a master regulator of glycolysis. And so you guys should be able to start speculating right now on why that's a good place to regulate glycolysis. Do you guys remember what we talked about in chapter 15? You probably not, because you're all still traumatized from preparing for your exam. So we talked about how we want to regulate metabolic pathways at steps that are pseudo-irreversible, meaning that they release a lot of energy. And so the way I draw those arrows is correct. It would be hard to go backwards. It's hard to go from fructose 1,6-bisphosphate to fructose 6-phosphate using this enzyme, because we would have to get a delta G that's negative, we would have to have a fructose 1,6-bisphosphate concentration that's so high that it's just not realistic. So that's a good way, that's a good reason to regulate there. But then you'd say, well, okay, but you're also releasing a lot of energy here. Why wouldn't you regulate here? And that's going to come down to what we had talked about, where we said that sometimes you don't want to regulate the first reaction of a pathway, because very well may end up being the case that this step right here is not only important for glycolysis, it might be important for other metabolic pathways. In other words, is glycolysis the only pathway that's utilizing glucose 6-phosphate? Alex is shaking his head. He's like, no way. What's your last name, by the way?
Velasco.
That's what I thought. Yeah, okay. I have too many Alexes and Chases, and it's hard to keep track of all y'all. See, but I am trying. And so yeah, maybe we can regulate here to some extent, but step 3 is going to be really where we want to regulate. And that's because once we commit to generating fructose 1,6-bisphosphate, that's where we really are committing to doing glycolysis, and we're going to have to, once that molecule is generated, we're going to have to finish this thing up. We're going to go all the way to the end. All right, so once we get to fructose 1,6-bisphosphate, we're actually going to cleave that into 2 different molecules. Molecules that you guys have heard the names of, because we've done some homework questions with them already. Dihydroxyacetone phosphate, or as I like to call it, DHAP. So much easier. And glyceraldehyde 3-phosphate, GAP. Okay? Now, once we get to those stages, we are now entering stage 2, where what's going to happen is we're going to have the coupling of energetically favorable reactions, that is the oxidation of we'll see shortly, of GAP, which is going to generate a molecule with high phosphoryl transfer potential. Glyceraldehyde 1,3-bisphosphate, right? And so that glyceraldehyde 1,3-bisphosphate, what did I call it? I said something wrong. 1,3-bisphosphoglycerate. Wow, okay. I would rather just call that 1,3-BPG, right? Just like that. And you don't have to memorize so much. Yeah. That 1,3-BPG was in table 15.1, and I even traumatized you a little bit in putting it on your exam. It's an important molecule. If you remember, the free energy of phosphoryl hydrolysis of 1,3-BPG was greater than the free energy of hydrolysis of ATP. And that meant that it would be energetically favorable for 1,3-BPG to transfer one of its phosphates to ADP, generating ATP. And so that's exactly what we're going to see at this step. I do have to mention, before I get there, I was too excited. And so glyceraldehyde 3-phosphate and DHAP were generated in the last reaction in stage 1. Now, DHAP is not a useful molecule when it comes to this metabolic pathway. GAP is going to be the one that's going to be utilized by the next enzymatic reaction in glycolysis. But the good news is that DHAP can be isomerized into GAP by this enzyme triose phosphate isomerase. And so we're going to do the molecular mechanism. We're going to look at the structure of triose phosphate isomerase. It's a very famous enzyme. And we're going to look at the residues in the active site, and we're going to ask those questions. We're going to say, how are those residues catalyzing the reaction? And then you guys are now going to start seeing why I keep making such a big deal about acid-base catalysis and manipulation of the pKa's of those groups that enable catalysis to occur. So once we have GAP, whether it was generated directly or indirectly, that GAP will get oxidized. And that oxidation is going to be so energetically favorable that we could actually do stuff with that energy, useful stuff. We're going to be able to take inorganic phosphate and attach it to GAP upon the oxidation of GAP. And that's going to generate the 1,3-BPG that's going to ultimately be utilized to generate ATP from ADP and orthophosphate. And so we're going to study the mechanism of action of that enzyme, glyceraldehyde 3-phosphate dehydrogenase, because this is a case study in how you can have an enzyme that figures out a way, a molecular mechanism, to take the free energy of oxidation of something that's reduced, store that energy, and then utilize that energy to generate a molecule of high phosphoryl transfer potential, 1,3-BPG. I'll give you a hint. It is going to involve the formation of a thioester bond between the enzyme active site and the substrate. So we can think of that as an enzyme intermediate, an enzyme acyl intermediate, and that intermediate is going to be storing our energy. Okay? So if that doesn't make sense to you, that's fine. We're going to look at that in great detail. I'm just giving you the introduction today. All right, so after 1,3-BPG transfers its phosphate to ADP to generate ATP, I want to point out, why are they putting 2 there? They're putting 2 there because for one glucose, we're ultimately going to get 2 molecules of GAP. Right? We get 1 GAP here, and then the DHAP is converted to GAP. So that's why they're saying, hey, we're actually getting 2 ATP out of that 1 glucose. Then what's going to happen is the 3-phosphoglycerate is going to be converted to 2-phosphoglycerate by what they call a mutase, but we could also think of that as an isomerase, group transfer reaction. And then the 2-phosphoglycerate is going to be converted to phosphoenolpyruvate, which, I don't know if you remember this, but if you consult table 15.1, that was the molecule on top of the table. Right, so of all the biological molecules that we're discussing that are phosphorylated, that molecule can transfer that phosphate to any of the other molecules in our table in an energetically favorable fashion. And so that's why when phosphoenolpyruvate is converted to pyruvate, we're going to see the generation of ATP in an energetically favorable manner. Not surprisingly, this is catalyzed by an enzyme called pyruvate kinase. Why is it a kinase? Because it's transferring a phosphoryl group to a molecule. So it's phosphorylating a substrate. Bless you. Look at your fancy hairdo. Sorry? I mean, learning how to have your mojo on this stuff is a little tricky. I mean, as you know, I'm not a huge believer in brute force memorization. That being said, in this case, it probably would be beneficial for you, especially those of you who are going to be taking the MCAT or DCAT or whatever CAT. KCAT. All right. So now we're going to go through these things in some more detail. So the first reaction is the phosphorylation of glucose, and it's catalyzed by hexokinase. See, Alana, I did that by peer memory. So as we already discussed, as I already alluded to, that phosphorylation is important because it's basically setting us up to generate fructose 1,6-bisphosphate, right? Which that molecule is important because when it gets cleaved, we're ultimately going to get 2 GAP molecules, and hopefully you guys now see that GAP it's a great molecule because it can get oxidized to form 1,3-BPG, and we're going to get ATP ultimately out of that. But there's another good reason. When you and I consume carbs, and it goes into our digestive system, enters our blood, and then it goes around to the various cells of our body that want it. We talked about brain, but also our muscles store glucose, a lot of it, actually. And so what you don't want is to go through all that work of putting glucose in the cells only to have it diffuse out, right? Because unfortunately, we're still beholden to the laws of physics here. And just because we want glucose to stay in the cell doesn't mean that it will. The laws of thermodynamics are still applying, meaning that it would be energetically unfavorable for a cell to have a higher concentration of glucose than, say, the outside of that cell. So the solution to that is to phosphorylate that molecule. So when we have glucose 6-phosphate, it can't get out of the cell. Now, these things don't just diffuse across the membrane. We're not going to get into this in great detail, but glucose is too hydrophilic to get across a membrane on its own, so it needs proteins that act as transporters. Now, at that point, you guys should be able to tell me exactly how those transporters are working. You would say, well, they have to have a binding site for the glucose, and that binding site has to rely on the formation of weak force interactions between glucose and the protein that are stabilizing. And so you're going to imagine, you're going to say, well, for that to occur, there has to be this binding site that has the perfect shape and physical chemical properties, the complementarity, just like you all said on the exam when you're talking about binding sites on proteases. The same exact thing. And so if I put a phosphate now on the OH group that is attached to carbon 6, it's not going to fit anymore. And even if it did somehow fit, I now have this negative charge that I'm placing into this binding site where it's probably not going to be complementary. In fact, it's probably just going to disrupt binding. So that's why glucose 6-phosphate is now stuck inside of our cells. It can't bind anymore to these transporters, and that's how our cells are able to maintain a nice, healthy, where our cells are like glucose squirrels, where they're just going to be, working hard to stash a bunch of glucose in the cell, and once that gets phosphorylated, it's not going anywhere. Okay, oh, look at that, they did the arrows right this time. Thank you so much. So it's going to prevent glucose from leaving the cell. Your book also says that it destabilizes glucose. I'm not a big fan of that. I mean, I'm sure it's true. We're not really, that's not what I think is important. What I think is important is to understand that we're just setting this molecule up so that we can generate GAP. Because GAP is the money molecule. GAP is the molecule that's going to get oxidized to generate 1,3-DPG, and we know we're going to get ATP out of that. And then after a few more steps, we know we're going to get phosphoenolpyruvate. We're going to get more ATP out of that. And we're also going to generate pyruvate, which is a precursor to acetyl-CoA. So that's the way that's how I think about it. I mean, this phosphorylating this molecule is putting us on the pathway to getting the goodies. I mean, another thing I want to point out is that GAP stands for glyceraldehyde and 3-phosphate. So glyceraldehyde 3-phosphate has a phosphate, right, at the 3 carbon. And I'm going to generate ultimately 2 of these molecules from 1 glucose by going through the first 5 steps of glycolysis. And so where do those phosphates come from? They have to come from somewhere. This is where they're coming from. That's how I would think about this. Okay. Do you guys remember, have we discussed induced fit? I think when we were in chapter 8, we went over it very briefly. So now I want to really hit this. So induced fit is a mode of binding between an enzyme and a substrate where there's going to be 2 conformations. One of those conformations of the enzyme is not going to be in the proper orientation to bind to the substrate, whereas the other one is. And there's a little bit of the magic that occurs that when substrate is round, it can induce this conformational change. That's what they mean. And I don't really want to try to get into any more deeper of an understanding. I mean, you guys understand that these molecules can't talk to one another, and the substrate's not saying, hey, you, change the conformation, right? So you have to use your imaginations to some extent. So what they're showing us here in this figure is a C-alpha trace, so a backbone trace of hexokinase in 2 different conformations. The conformation in light blue is going to be the conformation that exists in the absence of glucose. And then the conformation shown in magenta is going to be the conformation that exists in the presence of glucose. So I think what you can see is that this is a little bit like a Pac-Man, where we have our Pac-Man being kind of open. This glucose binding site here. And then we're going to have Pac-Man closed. And so I think we can imagine that in the open conformation, and the residues that interact with glucose are still there, they're just not in the right orientation to actually interact with all of those OH groups that are going to form weak force interactions that stabilize the enzyme substrate complex, whereas in the closed one, they are. So if you look at this picture, I think it's pretty easy to see that, first of all, there's 2 domains. So this one really is not moving, but this upper one is. And I think we can see that in the glucose binding site, all of these residues here that are on the domain on the top in the magenta structure are much closer to glucose. So you can imagine what that would look like. You can imagine that if we could see the side chains, we would see side chains that are approaching hydrogen bonding distance, salt bridge distance, and van der Waals distance to our substrate. And so only in the presence of glucose does this occur. And now we have to kind of ask the question, why does this happen? There's got to be a reason why this evolved. And the reason is because this reaction is ATP plus glucose becomes glucose 6-phosphate plus ADP. I want you guys to imagine what would happen if we just had ATP bound without glucose. And if that active site were to form, it's easy for us to imagine that, first of all, I want you guys to imagine how this reaction is occurring. And at least for me, I've got my sugar, and this is not, I'm going to do my best, but sorry. Something like this. I think this is right, then there's some other stuff. So if I have phosphoryl transfer to ATP, this will just be a bunch of other stuff. Can anyone tell me, what do you guys think the enzyme is going to need to do to promote this reaction? There's a few things that come to my mind. Seth?
It's going to have to break off the phosphate group from ATP.
No, I don't agree with that. That could be a mechanism, but based on what you know so far, I'm thinking in a different direction. Shalta. Yes. Shalta. So we need to put a powerful base next to that OH group so that we end up with powerful nucleophile, which can then go ahead and attack that phosphate. This is very similar to the reactions that we discussed in chapter 9, right? Activation of a water molecule, same thing. In fact, it's an OH group. It really is the same thing. It's just, in this case, the OH group is on glucose. So what would happen if instead of sugar binding there, if there was a water molecule? And you guys are probably like, well, oops. You're like thinking, well, why would a water molecule bind where glucose is binding? Well, because the active site of the enzyme is going to form probably a weak force interaction or multiple weak force interactions with that OH group. And it has the powerful base there to help deprotonate it. So in reality, it is reasonable to assume that a water could eventually bind there. And maybe if the affinity of water is not so great, we can imagine that would be made up for by the fact that the concentration of water in the cell is 55 molar, which is ridiculous, right? The concentration of glucose is maybe 1 millimolar. So that's, 1,000 times 50 is 50,000, right? It's a huge difference. And so this is why, the primary reason why we observe induced fit substrate binding in so many of these reactions. Because if a completed active site was always present on the enzyme, then in the absence of glucose, but in the presence of ATP, this enzyme becomes a really efficient ATP hydrolysis machine. And that would be bad, right? that would be just burning our precious fuel for the sake of burning it, and that's not efficient. All right, so induced fit is a model, it's a mechanism for ensuring that the active site only forms in the presence of substrate, the presence of glucose. Then they also mention that the active site is hydrophobic, which favors formation of the transition state, and so you guys can imagine why that's advantageous, because it's going to involve manipulation of pKa values of important ionizable groups. But I don't really buy this reason nearly as much as the first one, because it absolutely is true that even without induced fit, an enzyme can have an active site that is hydrophobic. So for me, it's all about ensuring that you only create an active site in the presence of substrate, so we don't end up expending our precious ATP. Okay, so step 2, isomerization of glucose 6-phosphate to fructose 6-phosphate is going to involve opening up this ring form, and we'll have this open chain form. And then this is going to get isomerized to this. So you can see the major difference. We have to do a little bit of chemistry talk here. Sorry, the way it is. This is called an aldehyde. This is called a ketone. So this sugar is called an aldose. This sugar is called a ketose. So through a mechanism that we won't discuss, I don't think we do at least, the aldehyde is going to be converted into a ketose. I can tell you, though, this involves a ton of general acid-base catalysis. And for those of you who want to see this mechanism, you know what to do in office hours. And then once the ketose is formed, it can then close back up, forming fructose 6-phosphate, as shown here in this 5-member group 1. Okay, then in step 3, we're going to observe catalysis of another phosphoryl transfer to generate fructose 1,6-bisphosphate, which I told you is a really, really important reaction, and this enzyme is going to be heavily regulated. You guys should be thinking to yourself right now, how is that, what are the mechanisms of regulation? You might think right off the bat, you'd say, maybe this enzyme is an allosteric enzyme. Maybe it has allosteric effectors. Maybe it binds to the binding cooperative. Maybe it's modified by phosphoryl groups itself to modulate its activity. Yeah?
What is the rate determining step?
I don't know if this is rate determining. That would just boil down to how good is this reaction at lowering the activation energy barrier, which we don't have any information for. So then here is the reaction catalyzed by aldolase. It's going to cleave between C3 and C4. We're not going to worry about the mechanism, generating GAP and DHAP. That's simple enough. All right, so now we get to this reaction that I told you that we're going to actually get into enzyme mechanism today. And so DHAP, or dihydroxyacetone phosphate, is not useful in the sense that we just don't have enzymes that can convert this molecule into 1,3-BPG, which is what we want, right? But we can easily isomerize DHAP into GAP. So let's take a look. So you can see that what's going to happen. One thing I want to point out is that both of these molecules have a CH2O-phosphate. That's totally identical. So really what's happening is we're going from a ketose to an aldose. So somehow this enzyme is going to take a molecule that has a ketone group, and it's going to figure out how to change that into an aldehyde. So in a lot of ways, I think if you understand this, you'd probably be able to speculate on what the mechanism is for the fructose, for the isomerase that converts glucose 6-phosphate to fructose 6-phosphate. All right, so here is the structure of the enzyme called triose phosphate isomerase, or triose. It's called TIM-BARREL. I'm actually not even 100% sure what the TIM acronym stands for now. That's okay. What is very apparent about this enzyme is its structure. This is an extremely common structure, guys. So I want you to first notice that we have a beta sheet in the middle. And that beta sheet is actually a barrel, meaning it's fully closed. Do you guys remember when we talked about beta barrels? Or we said it's basically a beta sheet that's folded around like a piece of paper such that all of the beta strands are hydrogen bonding to one another. And then on the outside are alpha helices. And so this is commonly referred to, I believe, as an alpha beta barrel. And I want you guys to understand that all of these secondary structure elements have to be connected somehow. And that is commonly done through the action of what we refer to as loops. These are segments of the polypeptide chain that connect secondary structure elements. So whether that's connecting, I don't know how easy it's going to be. So here's a beta strand, and we can see that here's a loop going all the way here. And then it goes to this helix. That has a loop that goes to this beta strand, and then that has a loop. Boy, that's not super easy to follow. Eventually getting to that helix. So we can kind of speculate and say probably the way this structure works is we go from a beta strand to a loop, to an alpha helix, to another loop, to the beginning of the beta strand, and then the end of that beta strand has a loop that connects it to the end terminus of the next alpha helix, and so on and so forth. And so what's going to happen is we can think of this thing as kind of like a donut. This is our triose phosphate isomerase, or a tin barrel. And so we've got these helices on the outside. And what ends up happening is you end up in the center with a bunch of loops like that. And that's going to be our substrate binding site, or I could also easily say our product binding site, right? Substrate's going to bind here. And so basically, that's one of the reasons why this is such a successful fold, because the fold creates a binding site and an active site simply by all of these loops that meet in the center of the donut. Okay, sorry. I like maple bars, I like chocolate, I like sprinkles, I like old fashioned, I like it all. All right, so then they're going to show, so they're showing us the substrate, So again, that substrate can be either GAP or DHAP. So I immediately look at this thing and I go, what is that group down there? Anybody? It's the phosphate, right? So, and they're deciding to show me two residues, glue and his. Now, these are residues that have become good friends of ours, right? Histidine is maybe Dr. K's favorite residue. Why is it Dr. K's favorite residue? Anybody? What? What?
Very versatile.
I want more specifics now. You guys are big boys and girls, big biochemistry boys and girls. Has a pKa of 6 without doing anything. And so in this environment of the active site, the enzyme is not going to have that hard of a time promoting an environment that is going to promote proton transfer, both to and from the substrate. What about glue? Same thing, right? So this is going to be a bunch of acid-base catalysis. All right, so here is the mechanism. Once again, we messed this up. Dr. K rescue. So we're starting with DHAP, but we could totally do this with GAP, and we will. So here is DHAP, and basically we've got our histidine over here, one side, and our glue on the other side. And so the first step of the reaction is going to be deprotonation of C1. What do you guys think is the pKa of C1, the carbon-hydrogen bond at C1 in DHAP? Do you guys think that's going to be super acidic? Is that hydrochloric acid, or is that the opposite? Well, it's definitely not hydrochloric acid.
Acidic enough to give protein.
Well, throw out a number. Tell me what you think. First of all, tell me what you think the pKa of a carbon-hydrogen bond is in water without the enzyme.
Depends on oxygen level there.
It definitely is going to depend on what's here, but just give me throw out a number.
32.
I like 32. You said 32. Oh, hey Tyler. Yeah, I think that's a good number. Super, super high. Why is that high? Because, oh boy, am I going to have to try to remember how to draw DHAP from memory? Let's see. I'll cheat a little bit. So I've got this. I've got two items there. Okay. And I have a ketone. And I've got phospholipid. That. And then we're going to have our OH. That looks good. So if this thing were to spontaneously deprotonate, I'm just going to draw this way right now. Look at what I've got now. I've got what we call a carbanion. Do carbons like negative charges, or positive charges for that matter? They hate it. They hate it. So that's why this has a pKa, at least in the 20s, if not Tyler's 32. So on its own, this is just not going to happen. It's just so energetically unfavorable. So we have to ask ourselves the question, what is the enzyme going to do to promote this? What it's going to do is it's going to put a strong base right next to that CH bond. So that acid is going to be in an environment where it is happy to be protonated. So the deprotonated form of glue 165 is going to be stable. The protonated form is going to be stable. So in other words, the pKa of this guy, of glue 165, we're going to guess it's going to be around the value of the pH, because as we're going to see shortly, we want that not only to be able to pick up a proton, but to donate a proton. So once DHAP is deprotonated, what's going to happen with that yucky carbanion? It's going to get stabilized because our histidine at position 95 is going to donate a proton to form what they call an ene diol. The ene part means carbon-carbon double bond. The diol is referring to the two OH groups. And so that's how this is going to get stabilized. We're going to immediately protonate this thing so that the electron cloud is delocalized. Right? So we don't have a negative charge, formal negative charge, squat on that carbon anymore. Now I want to point out something that's weird that you guys probably have never seen before. Does that look normal to you? Yeah, Jacqueline's like, So what do you think's wrong with that? Huh? What? Yeah, it's doubly deprotonated, exactly. So what we've been talking about so far, and your book doesn't even talk about this, by the way, so heads up. When we talked about histidine, I want to go to black. We've been really only talking about the ionization of this form. Proton. And this is the one with Schalge's pKa of around 6. So what they're showing me is another deprotonation event. And we don't even know what the pKa is of that. It's not even in your book. Kind of annoying to me. I had to look it up. When I first started teaching Biochem 1, I saw that, I was like, what the heck? What's going on with that? So I looked it up. I'm going to let you guys finish writing, and then I'm going to go back to my notes. So this looks like a Wikipedia page. And so imidazole is amphoteric. That is, it can function as an acid and a base. As an acid, the pKa is 14.5. There's my answer right there. Yowzers. 14.5, that's lousy. This pKa, 14.5. So if we go back to the slide here, how is this possible? How is this ridiculously crappy acid donating this proton to stabilize my unfavorable ionization event on C1? Can anyone tell me, please? You all know the answer, by the way. You really do. Microenvironments. Who said that? That's right. Yeah, the pKa in the active site is not 14.5. I can tell you that. It's probably close to the pH. Because as you guys are going to see, the mechanism of of this reaction, of the isomerization of DHAP to GAP, or of GAP to DHAP, is a series of reversible proton transfer events. And so that means that the acids and the bases in the active site have to be equally happy in the protonated state as in the deprotonated state. So what I mean by that is, how do I get that to be roughly equal to 1? Well, you guys know the answer. When the pKa of the group is roughly equal to the pH, 10 to the 0 is 1. So we know that even though the carboxylate on a glue side chain has a pKa of around 2 in water, it is definitely not going to be 2 in the enzyme. Vlad's smiling because he's like, I get this. And then, same thing with this crazy histamine. Even though this looks weird and messed up, I know I am right. I know that the pKa of that acid is tweaked by the enzyme so that it's going to be perfectly happy to be both donating a proton and accepting a proton. So once again, here we are seeing principles that we've been discussing all semester long in action. All right, so now, once we have the enediol, two things can happen. The boring thing that can happen is our His 95 can abstract this proton here. Well, if that were to happen, if this were abstracted, then these electrons would come back in here, reforming the ketone. And then the electrons in the carbon-carbon double bond would pick up the proton from the glue side chain, and we'd be back over here at DHAP. On the other hand, if the histidine grabs a proton from the other OH group, now what's going to happen is that lone pair of electrons on the oxygen will come in, reforming a carbon-oxygen double bond, and these electrons still pick up the H. So let's go over here and do that. Well, I'll tell you what, I'm just going to draw my enediol. Let's see if I can do this without messing up. You guys will tell me if I mess up. That looks good to me. I'm going to have my imidazole grab proton from this OH. That's going to come in, form a carbon-oxygen double bond. And then carbon 1 is going to get protonated, and it's going to get the proton. It's basically, the glutamic acid is only borrowing this proton. It's not taking it away permanently. The substrate's going to get it back. And so at this point, we're done. Now we're going to get a molecule that looks like this. And that's GAP. I want to show you guys how I can go backwards. So let's get a new page. Let's write GAP. And then I won't cheat, I promise. So if I mess up, I just mess up. It's okay. It happens. I want 2 protons there. I totally drew that backwards. Well, like I said. Yeah. No big deal. And so I know I'm going to deprotonate this sucker, right? That's how I'm going to form that enediol. And so the question just becomes, which proton am I going for? We know we're not going for here, because the CH2O phosphate is common between GAP and DHAP. So it's either going to be this one or this one, and the free energy of deprotonating on the aldehyde is different than the free energy of deprotonating here. And so I know that what has to happen here, I know that I'm going to deprotonate at C2. I'm going to deprotonate on the carbon with the OH. And then deprotonation is going to cause the ketone oxygen to pick up a proton from his. It's going to generate COOH. It's going to generate our enediol. And we have his minus. And then you go back to DHAP. I know that I'm going to abstract proton now from here. The opposite one. So obviously where I'm placing the histidine is not in 3D. Deprotonation of the OH group in C2 will cause the electrons in that OH bond to now form a C double bond O. And then the electrons in the carbon-carbon double bond are going to pick up a proton from my glutamic acid. And I'm now at DHAP. And I want to emphasize to you guys that the enzyme doesn't have any control over this reaction. In other words, the enzyme can't say, oh, I bumped into GAP. What a shame, I'm just going to get rid of this GAP. That's not what happens. If the enzyme happens to bump into GAP, it's like, you know, that awkward situation of bumping into your ex. Ugh. Okay, let's get this over with. And so the reaction will go backwards when the enzyme bumps into GAP or DHAP. It's just the thermodynamics that will guide what the relative amounts of GAP and DHAP are in the cell. So in my last 2 minutes, just please bear with me. And so the enzyme is just going to catalyze the interconversion between DHAP and GAP using the mechanism that I'm showing you. But it has no control over what we really want, which is DHAP to be converted to GAP. But we have an ace up our metabolic pathway sleeves, because GAP is going to be converted into 1,3-BPG in the next step of glycolysis, right? So GAP levels are always going to be kept really, really, really low. So let's write that out. The reaction is DHAP, GAP, and the free energy, the delta G naught prime, around 0. But if GAP levels are low due to the next step in glycolysis, the delta G will be negative, right? And so that's how metabolic pathways work, right? It's not the enzyme that is somehow distinguishing DHAP from GAP. Instead, the metabolic pathway is going to keep the levels of GAP low relative to DHAP. And then at that point, it's just more likely that the enzyme will bump into DHAP because there's more of it around. All right, that's it. See you guys at office hours at 1.